Okay, so this is roughly where we got to yesterday. So and look at those photosynthetic pigments. I'll do a quick recap of those because just sort of started them at the end of yesterday. Look at how we investigate them, quick recap of ATP, and then start to actually look at that third stage of photosynthesis, which is that light-dependent reaction. So, as I said yesterday, a photosynthetic pigment is a coloured, that's a really important part, it's a coloured biological compound, and we find it either in chloroplasts or in photosynthetic bacteria. So, like we said, some bacteria are able to photosynthesize. And it has to be able to capture light energy for photosynthesis. So plants, we have two types. We have of photosynthetic pigments. So we have chlorophylls, which are those green ones that we see in plants and that we talk about a lot being present in chloroplasts. And we also have carotenoids. I was saying yesterday that mentions it's very similar to carrots, the word, and that's because that's the pigmentation that we find in carrots as well, that gives them that orange colour. So the reason these photosynthetic pigments are coloured is because they absorb particular wavelengths of light and are able to reflect others. So if we think back to basic physics, different colours absorb different wavelengths of light. So we sort of categorise different wavelengths of light as being in different coloured zones. So they're able to absorb some and then they reflect others. So chlorophyll is that pigment that gives plants their green colour, and it gives it this green colour by reflecting the green light. So if we think back to, again, early physics at school, the colour of the object that you see, that's the colour that it reflects, and then it absorbs all the other colours. So green reflects anything that's green coloured, such as chlorophyll, reflects green light, whereas carotenoids reflect red, orange or yellow light, hence they give carrots that orange colour I was talking about. So that's basically what a photosynthetic pigment is, able to absorb certain wavelengths of light and reflect others. So something you have to be able to talk about, and they might ask you about in an exam, is actually being able to look at how we might look at the effect of different wavelengths on photosynthetic pigments. So it's quite a basic experiment, really. If you think back to GCSE, and you'll have done one very similar that was looking at light intensity. So you would have a plant that does photosynthesis. Usually we use pondweed because it's an aquatic plant. We can measure its rate of photosynthesis quite easily because as the oxygen is given out, it produces bubbles, and you can either count the bubbles or you can collect that gas in a gas syringe. And we would move the light further and further away and see what effect the movement of the light would have on the rate of photosynthesis. This experiment is actually very similar. However, we'll look at the effect of wavelength of light. So this time it's not light intensity, so we're not going to move the light, but we're actually going to change the wavelength. And so basically we'll just have a quick look at how we would do that. So basically, step one, we would get light of a particular wavelength and we would shine it on the pondweed. The rate at which the oxygen is given off is used as the measure of a rate of photosynthesis. So basically, exactly the same as what I was just saying. We get some pondweed, put it in a beaker, put a funnel over it, attach a gas syringe to it. As photosynthesis starts to occur, oxygen is produced. This will then cause the gas syringe to fill up, and then we can measure the rate of photosynthesis to how much that gas syringe has, how much volume has been collected of gas in that gas syringe. So yeah, really basic setup of an experiment. And as you can see, we've got this purple sort of block in front of the light here. What this actually is a filter, and this will be a filter that we're going to use to get different wavelengths of light. So we put this filter in front of it, and we're going to measure the rate of photosynthesis. So we've measured the rate of photosynthesis in the highest at the wavelength of 400. So that's the first thing you would do. We plot it on a graph. Then step 2, you can probably guess, we're going to change the wavelength of light. So what we do to do that is we change the colour of the filter that we put in front of the light. That will change the wavelength that reaches the pondweed. We do the experiment again, exactly the same. So we leave it to do photosynthesis for the same amount of time, record the amount of gas that is collected in the gas syringe for the same amount of time, and then plot that onto a graph. So as you can see here, this blue filter has a wavelength heading towards 500, and our rate of photosynthesis has decreased here. Step 3, we're going to put another filter on. So for wavelengths between about 500 and 600, the rate of photosynthesis is really, really low. So I'm just going to pause it here. Little or no oxygen is collected in the gas syringe, and little or no bubbling is observed. So I'm going to actually go back to here to where we were and play it again so we can see it. So put a new filter up, and as you can see, the gas syringe is hardly moving, and we've got really, really low rates of photosynthesis. We've changed the filter now, and we're heading towards 600, and it's still low, but we're starting to see that increase in the rate of photosynthesis. But again, it's still really, really low. Then from 600 to 680, the rate of photosynthesis starts to increase again, and we get more oxygen being produced. So hopefully, what we should see, new filter on there, higher wavelengths, and more photosynthesis is now going to start to occur. But we get 700 and it plummets again, which is really important to remember. So the amount of light absorbed by the chlorophyll can then be measured using a technique called spectrophotometry. There we go, spectrophotometry. It's an awful word to try and say. And there's a few ways we can do this. Basically, we can do this to measure the amount of chlorophyll coming through, and the results of this are plotted onto the same graph. So I'm going to get this up so we can see that. So the absorption of the spectrum of chlorophyll. So basically what you would do is in that situation, you would take a sample of the plant, and then we would use the absorption rate to look at that. As you can see, when we plot them on the same graph, we've got a very similar pattern. The amount of absorption that's actually happening in the chlorophyll compared to before, because just because the rate of photosynthesis is happening doesn't necessarily mean, it doesn't show us exactly how much light is actually being absorbed. So this is actually looking at how much percentage of light is actually being absorbed. So we can see that around 400 it's very high, 700 it's high, and everywhere else it's very, very low. You don't need to know the actual process of how we do that, just that we are able to do that. So, quick questions about photosynthetic pigments. These sort of questions you might get asked, that they like to ask you to maybe try and trip you up. So what is the best way to measure the rate of photosynthesis of a plant when it's exposed to different wavelengths of light in an experiment? And that's a really key part here, is this bit, different wavelengths of light. They like to ask questions like this because actually lots of people think back to a GCSE experiment and go, oh, we move the light. And we don't move the light in this situation. We're not looking for light intensity. We are looking for different wavelengths of light. So we're not going to measure the starch produced by a plant over time using iodine solution. That's not beneficial. We are going to measure the oxygen produced by a plant over time using a gas syringe. The other option is to measure the change in height of the plant over time using a ruler. That again would be completely unrealistic because that would take a large amount of time and depend on growth rates, and a lot of other different things would affect that. So, similar question again. In the same experiment, what's the best way to vary the wavelength of light? and that is to place different coloured filters in front of the lamp. It's nothing to do with the brightness of the lamp, and it's not different coloured lamps. Now, that's really important. So we need the same lamp every time. That's obviously to control the experiment, to make sure that nothing is changing. If we change the colour of the lamp, that can actually affect the temperature. And actually, it doesn't necessarily affect the wavelength. It can do, but it can be quite tricky depending on how they've coloured the light. So actually the best thing to do is we use coloured filters. It ensures that we aren't changing any other variables, we're just putting a different filter. Again, if we're going to control this really carefully, we're going to want to make sure actually those filters are made of the same sort of material and they're the same thickness. So ATP we've met a lot in the past, and we're going to do sort of a quick recap of it again now. So ATP, it stands for adenosine triphosphate, and it's really, really important molecule, and we find it in every single living cell. It's basically an energy currency. It provides energy for cellular processes. It can diffuse really, really easily, and provides energy for cellular processes. It's made up of three things. It's made up of adenine, oops, that started to move, a ribose sugar, and then 3 phosphate groups. And that 3 phosphate groups is what makes it a triphosphate. So adenine, the sign, adenosine, is that part comes from ribose, and then triphosphate, tri meaning 3. So ATP, this is made in the light-dependent reaction photosynthesis. So that's that first part of photosynthesis. And we actually make it from a molecule called adenosine diphosphate. As you could probably guess from the name of it there, adenosine triphosphate means we've got 3 phosphate groups. Adenosine diphosphate is exactly the same molecule, except for it only has 2 phosphates added to it, and it has 2 phosphate groups. So ATP is made from adenosine diphosphate and an inorganic phosphate group, which we always write as P, the little I, to show it's an inorganic phosphate. And basically these two are bonded together, and when they're bonded together, they make adenosine triphosphate. Now, this process requires energy. It can't just happen on its own. So whilst ATP is an energy molecule, we need energy to actually make it. ATP actually releases energy in the light-independent reaction. So in the second stage of photosynthesis, where light is not required, it releases energy. And that is when the bond between the inorganic phosphate groups is broken. So basically, this bond here is broken, and it will produce ADP and an inorganic phosphate group. So you can see that they can be joined together or they can be broken apart. So I'm going to look at that in a little bit more detail now. Actually, how do we join them? How do we bond these together? And how do we break these bonds? So in the light-dependent reaction, so this is the first part of photosynthesis, and we're going to look at that in a bit more detail in a bit, ADP and an inorganic phosphate, so this is our inorganic phosphate, bond together. If there is energy provided, we need energy, and it forms ATP. So adding a phosphate molecule is called phosphorylation, okay? You'll have come across phosphorylation before, and this process of phosphorylation happens in the light-dependent reaction of photosynthesis. And in this case, the energy source that's used to bond the ADP and the inorganic phosphate together is light. So because we use light, we actually call this process photophosphorylation. Photo meaning light, and then phosphorylation, that addition of a phosphate group. The opposite, when we break ATP down to form ADP, an inorganic phosphate, and energy, because actually that breaking of the bonds releases energy, which makes sense. If we require energy to build those bonds, actually when we break those bonds, we're going to release energy. To do that requires water, which you can probably recall means it's a hydrolysis reaction. So this is in the light independent reaction. So this is in the second part of photosynthesis. And ATP is broken down in the presence of water through a hydrolysis reaction, and therefore ADP, an inorganic phosphate, and energy is produced. Other thing that we use in photosynthesis is a new molecule that we probably haven't spoken about before. And you'll normally just see the shorthand of the letters that it used, which is NADP. NADP stands for nicotinamide adenine dinucleotide phosphate. May on occasion get asked to write that on an exam for a 1-mark question, or you may be given the initials and asked on a multiple choice which one of these for what does NADP stand for. It's a coenzyme that's involved in the photosynthesis reactions. So it's a dinucleotide, and it contains an adenine base, very similar to adenosine triphosphate and adenosine diphosphate. So it's got the adenine base here and a nicotinamide base. So that's why it is a dinucleotide, because it has 2 nucleotides in it, okay? The nucleotides are joined together through their phosphate groups. So here we go, we've got our nicotinamide here. We've all got a ribose attached to them. So nicotinamide with a ribose sugar, then the phosphate. As you can see, it's a basic base. And then we've got our adenine here attached to the ribose sugar and the phosphate, and the 2 phosphates join them together. Okay, so that's through their phosphate group. So we've got some bondings for bonding. So it's like that sugar phosphate backbone idea when we look at DNA. So in addition, in this molecule, we also have an extra phosphate, and that's really important to remember. And that's on the ribose of the adenine containing nucleotide. So we have this extra phosphate at the bottom. This extra phosphate is really, really important because NADP can accept electrons to be reduced to become reduced NADP. Rather than writing reduced NADP, you'll often see this written NADPH, and that means reduced NADP. It's H because these electrons, obviously, if you think about the electronic configuration, it works out the same as hydrogen. So we use the H for hydrogen to show that it's accepted some extra electrons. NADPH can be oxidized back to NADP and thus releases oxygen. Oxygen releases electrons. So remembering back to that basic chemistry, oil rig, oxidation is loss of electrons, reduction is gain of electrons. So reduced NADP has gained electrons, and then NADPH can be oxidized back to NADP and releases electrons. So which molecule is this? Quick look at it. Okay, well, we've got adenine here, we've got our ribose here, and we've got 3 phosphates. Therefore, it must be a TP, adenosine triphosphate. Which molecule is produced by hydrolysis in the light independent reaction? That's a DP, remember, because that's when ATP is broken back down into ADP. Which molecule is a photosynthetic pigment that reflects green light? Well, carotenoid is a photosynthetic pigment, but it doesn't reflect green light. It reflects reds, oranges, and yellows. ATP is an energy molecule, so it has to be chlorophyll. Which molecule is produced by hydrolysis in the light independent reaction of photosynthesis? And that is ATP. So remember, ADP and an inorganic phosphate bond together in the presence of light energy and make ATP. And chlorophyll is that photosynthetic pigment. I think this is, hydrolysis in the light independent. Why is it now gone back to the same questions before? What has just happened? It's like cycled back to the wrong question. Yeah, no, that's the same question again. Okay. Which molecule is produced by photophosphorylation in the light-dependent reaction in photosynthesis? Right back to the right question now. That is ATP. What molecule of this? Okay, so we've got adenosine here, we've got a ribose, and we've only got 2 phosphates. So it can't be ATP because that T stands for tri, which is 3. Chlorophyll is our pigment, so it has to be ADP. This is the light absorption spectrum for which molecule? It's not an inorganic phosphate, ATP, it is chlorophyll. Which molecule is sometimes described as storing energy because it reacts to release energy that is used to make complex molecules such as starch? That is ATP. So remember, when ATP is broken down during hydrolysis, it forms ADP, an inorganic phosphate, and releases energy. And finally, what else is produced when ADP is formed from ATP? That is an inorganic phosphate. So let's have a deep dive now and deep look into that first stage of photosynthesis, that light dependent reaction part. So we're going to look at what actually happens during this light dependent reaction. So if we think back to the chloroplast, the light, well, all of photosynthesis occurs inside the chloroplast. The light dependent reaction takes place in those thylakoid membranes of the chloroplast. So if we think about chloroplast really quickly, I'm going to draw one that's sort of been cut open a bit. It looks a bit like this. This is my bad drawing of chloroplast. And we have these stacks of thylakoids. So a stack of thylakoids is called a granum. And one flattened sac that's full of the photosynthetic pigment called chlorophyll is called a thylakoid. We've got lots of these stacks all over and they're joined together. And that is where the light dependent reaction takes place. So it takes place in lots of places, well, in lots of thylakoids all at the same time inside the chloroplast. Okay, let me try and get rid of. So situated in these membranes are light harvesting systems called photosystems. There are 2 types of photosystems. We have photosystem 1 and photosystem 2. And both have chlorophyll at their centres. Okay, you'll often see them referred to as just PS1 and PS2. And we'll look at them in a bit more detail in a second, okay? So don't worry about them too much. So if we look into the leaf in a little bit more detail, photosynthesis is occurring inside the leaf, inside the chloroplast, inside the thylakoid membrane. And in this membrane, we have 2 light harvesting systems. We have photosystem 1 and we have photosystem 2. And they both have chlorophyll at their centres. So this is our photosystem here. Light hits it and the chlorophyll absorbs that light. So what I'm going to do now is I'm going to talk it through to you once using the diagrams that are shown on here. And then probably going to recap it again using a different diagram that I'm going to draw myself and then maybe show it you with another one. Now, the reason why I'm going to show it you using lots of different diagrams is, again, like I've probably said loads of times, different textbooks, different revision guides will use different diagrams because basically you can use things that actually look like the parts of a plant, or people just often use flow charts of this because it's it's a reaction that we can use flowcharts to sort of describe. Now, again, this is something that you need to recall in detail, and actually being able to remember it as a flowchart is really important. So again, when revising, I think the best way to revise this sort of information is to write it down, write it in your own way that suits you. So it might be that you use a diagram. Some people do like step-by-step processes, but I think a diagram is really, really helpful, as in a flowchart type of a diagram for this process. And actually then you can come back to it later and you can try and draw it from memory. And if you've got bits missing, then you can look back at your original, add them in, and then keep practicing to get better. So the light dependent reaction has two parts to it. It has a non-cyclic photophosphorylation, and that involves both of those photosystems we just spoke about, photosystem 1 and photosystem 2. There is also a cyclic part of this photophosphorylation. So obviously, cyclic means it cycles, non-cyclic, it doesn't cycle. So non-cyclic photophosphorylation has photosystem 1 in it, photosystem 2, has 2 electron acceptors and 2 electron transport chains. So what happens is we actually start with photosystem 2. And photosynthesis, this light-dependent reaction, starts when light hits photosystem 2. So what happens is light comes along and hits photosystem 2 here. When the light hits photosystem 2, 2 electrons inside photosystem 2 become excited. These excited electrons are going to gain energy, and as they gain energy, they are accepted by that electron acceptor that is here. When they are accepted by that electron acceptor, they leave photosystem 2 electron deficient. Okay, so let's pause that and talk it through. So step 1, light. I'm really hoping light's going to be drawn onto here. Here we go. Light hits photosystem 2, and that's really important. We always start with photosystem 2. So light comes along, it hits photosystem 2, which has chlorophyll in its center. This light energy excites 2 electrons. These electrons gain energy, and they go to this electron acceptor. When they are accepted by this electron acceptor, this causes photosystem 2 to become deficient in electrons, and that's really, really important. So light energy hits, 2 electrons are excited, they get accepted by the electron acceptor, and photosystem 2 is now deficient in electrons. That takes us to step 2. Those excited electrons then come along and they pass, way too fast, along a chain of electron carriers. So we call that the electron transport chain. So here we've got those excited electrons that run that electron acceptor, and they pass along a chain of electron characters. So this is that electron transport chain. And as they're passing down this chain here, they start to lose energy each time they are passed on. As they lose energy, that energy is used. The energy that is released from these electrons is used to make ATP. And that is that photophosphorylation that we spoke about. So ADP and that inorganic phosphate receive the energy that has been released from those electrons along that electron transport chain. ADP and the inorganic phosphate are able to bond together, and therefore ATP is formed. So I'm just going to make this bigger, talk it through again. So the 2 excited electrons pass down the electron transport chain. As they pass along the electron transport chain, they lose energy, and that energy that has been released allows the ADP and inorganic phosphate to bond together using photophosphorylation to make the molecule ATP. And those 2 electrons now arrive at photosystem 1. So step 3. Light also hits photosystem 1. So it doesn't just hit photosystem 2, light also hits photosystem 1. And the same thing happens to the electrons. The electrons are excited. Again, it's 2 electrons. The electrons are excited and they pass to an electron receptor, acceptor. To replace them, photosystem 1 absorbs the 2 electrons originally from photosystem 2. So let me talk this through because I don't think this picture shows it very well. So those electrons that are on here at the minute, these ones here, haven't fully arrived at photosystem 1. So what's going to happen is light hits photosystem 1. There's some other electrons, another 2 electrons inside photosystem 1 that get excited. Same thing happens to them. They get excited, they gain energy, and they are accepted by the electron acceptor up here. Photosystem 1 is now deficient in electrons, but from the electron transport chain, those 2 electrons have just come down to photosystem 1, and photosystem 1 is able to accept those electrons that came from photosystem 2. So it's a bit of a recycling process here. Have I just skipped ahead? Yes, I have. So in the same way as photosystem 2, those electrons, again, are going to pass down another electron transport chain. This time, the energy that's released from them is used for the reduction of NADP to make NADPH, so reduced NADP. So the products of non-cyclic photophosphorylation are ATP and reduced NADP, and these are used in the light-independent reaction. So that's the non-cyclic part of photophosphorylation. We're also going to look at a cyclic part of it and then join the two together. So cyclic photophosphorylation involves just photosystem 1, 1 electron acceptor and electron transport chain. So it is actually still part of the cyclic and the non-cyclic, and I'm going to draw it out for you in a moment and break it up into the sections. But again, the process begins when light hits photosystem 1, and those 2 electrons that we spoke about earlier in photosystem 1 get excited and they are accepted by an electron acceptor and they leave photosystem 1 deficient. That is exactly the same as before, nothing has changed. So again, light hits photosystem 1, 2 electrons are excited and they are accepted by an electron acceptor. These electrons pass along a chain of electron carriers, and as they lose energy, they allow ADP and inorganic phosphate to bond together to form ATP. So the products of cyclic photophosphorylation are ATP, and this is used in the light-independent reaction. And then the last little part of this is the photolysis. So photolysis, lysis meaning splitting by water, photo meaning light. Water molecules are split by light energy, and this happens in the thylakoid lumen. So what happens here is light energy hits a water molecule, and with the aid of enzymes, the water is split into oxygen, hydrogen ions, and electrons. The hydrogen ions reduce NADP. So you know how we keep saying, oh yeah, we're going to reduce NADP in a bit. They're what we use to reduce the NADP to reduced NADP. which passes into the light-independent reaction. These electrons replace those lost from photosystem 2 in non-cyclic photophosphorylation. Otherwise, the photosystem 2 would just lose electrons, then it would never be able to be used again. And the oxygen is released as a waste gas. So here's an overview, and it looks really, really messy and really complicated. But this is our cyclic part. Oh, no, I've gone the wrong way. This is our cyclic part, non-cyclic photolysis. So before we do that, I want to get something else up. I just want the whiteboard. Why is that now really complicated? You just stop sharing the screen for a second to see if this works. Zoom's changed all of a sudden, and now I can't find where the whiteboard is. What I wanted was a whiteboard. It used to be really easy to get up. Don't want any of them. I might just have to get a white screen up. Where's the annotate button gone? I'll use this instead. I'll improvise. Share screen. Ah, whiteboard. There we go. I knew it would come up somewhere. So, change one place of where something is, I can never find it. Okay, so let's talk this. Let's talk this through again. So the light-dependent stage of photosynthesis, we start with PS2, so photosystem 2, so PS2. In PS2, we have those middles that have got chlorophyll in them, and they have those electrons in them as well. What happens is light comes along, and light hits photosystem 2, it's not light, it's photosystem 2. Those electrons are excited, and what happens to them, I'm going to do them in blue, is they gain energy and they go up to an electron acceptor. So here is our electron acceptor, electron acceptor. Okay, so we go up to the electron acceptor. So these are our two electrons. They get excited and they go up to the electron acceptor here. Once they arrive at the electron acceptor, they pass down the electron transport chain here. The electron transport chain. I'm going to actually write that somewhere else. I'm going to write it here. That's our electron transport chain. So as these electrons are passing down the electron transport chain, they lose energy. Okay. So as they lose energy, it means this can happen. We've got some ADP and we've got some inorganic phosphate. They can join from the energy provided by those electrons that are losing energy down the electron transport chain. Let's pop that here, that's our energy. And they make ATP. So that is that first part. Then photosystem 1 gets involved. So we've got PS1. And in PS1, we've got these electrons here again. These electrons get excited by light energy hitting them. And they rise up and they go up to an electron acceptor. Again, it's the 2 electrons, they're going to rise up and they go to the electron acceptor. Now, remember, they are replaced by the ones over here. Okay, so they're replaced by the electron acceptor ones over here. So the electrons that have come from PS2 replace the ones that are lost in PS1. So that makes it cyclical, partially cyclical. Then they go down another electron transport chain. And when that happens, ETC, we can have our reduced NADP, or our NADP is able to be reduced to reduced NADP. So I'll write NADPH here. And that occurs because of this. Okay, so we've spoken about it being cyclical and non-cyclical. So the non-cyclical part is everything I've just drawn. And then we have that cyclical part here. And it's basically, we call PS1 cyclical because what could happen is, and I'm going to draw that in green, this section here is cyclical. Because it's constantly having the electrons replaced via this electron transport chain here, every time 2 electrons are lost up here to this electron acceptor, a new 2 are replaced to it here. So that's why that is cyclic. This whole information that we've drawn here is non-cyclic. This does not cycle. And that is, oops, I've just moved the whole screen. What am I doing? That is non-cyclic, and that is non-cyclic because those electrons cannot be replaced down here. Okay, so that can't just keep happening by itself. So there's one extra bit that we haven't added on here, and that is photolysis. So let's do photolysis down here, and that happens at PS2. So here we have water, and she's split apart by that light energy that occurs here, and it produces 2 hydrogens and water. These hydrogens are then used here to reduce that NADP. Okay, so that's where they come from. So I'm going to get another diagram to explain it to you again. And that bottom bit, again, I'm just going to label that. This is photolysis. And this is just the first stage photosynthesis, the light dependent reaction. So move that down. Let's pop this up. Zoom. Wants to work now. Share screen. There we go. Here we go. Share. That's a chloroplast. Just want the diagram. So quick overview, like at this stage, it's just a different outline of it. So remembering PS2, PS1, these are all found in the thylakoid membrane. Stromas on the other side, thylakoid space on the other. So light comes along and they are absorbed by photosystem 2. Light comes along, hits photosystem 2. This excites those 2 electrons which are displaced They're raised to a higher energy level and they are taken in by an electron acceptor. So there are excited electrons, they go to the electron acceptor. They are picked up by electron acceptors and they pass it along that electron transport chain. The energy is down to the photosystem 1. The energy that is lost by those electrons as they go down that electron transport chain is used to convert adenosine diphosphate and an inorganic phosphate to ATP. So we make ATP and those 2 electrons have arrived at photosystem 1. Photons of light again are absorbed by photosystem 1. They excite another pair of electrons, which is then picked up by another electron acceptor. There we go. There we go. They're being excited. Now remember, there are another pair of electrons, or a different pair of electrons. And PS1 becomes unstable because it's lost 2 electrons and it gains them back from the splitting of water in the photolysis. There we go. Second electron acceptor, which received the electrons from photosystem 1, passes the electrons to hydrogen ions where they reduce NADP. And NADP is needed for light-dependent reaction. So, here we go again. Cyclic part, because we can keep replacing the electrons from here that go to there. Non-cyclic photophosphorylation, photolysis. So the products of a light-dependent reaction that pass into the light-independent reaction that pass into the second stage are reduced NADP and ATP. Products of the light-dependent reaction that then leave the plant and we don't use again during photosynthesis are oxygen. And products of a light-dependent reaction that are reused in another part of a light-dependent reaction are those hydrogen ions and electrons. So they're what allow that cyclical part to happen. So those hydrogen ions are used to reduce the NADP. And then we have those electrons as well that keep getting reused. So that's what happens with them. So the light-dependent reaction, we're not going to do this now. We're going to actually have a quick look at the light-independent stage, sometimes called the Calvin cycle. So, light-independent stage, it takes place in the stroma of the chloroplast. So remember, the stroma is that amorphous liquid gel, and it's full of enzymes. And the light-independent reaction, sometimes called the dark reactions because we don't need light, or you'll regularly see this, it's referred to as the Calvin cycle. And it's great because we can literally draw it as a cycle. It is cyclical, it is in a circle that we can learn. This reaction is actually consisting mainly of a cycle of enzyme-controlled processes. And it actually makes really complex molecules such as carbohydrates. So this is why it takes place in the stroma. We don't need the light. We've got the reduced NADP and the ATP from the light-dependent stage. And now we're going to use those enzymes that are present in the stroma to do this second final part. So again, we're in the leaf, we're in the chloroplast, but this time we're in the stroma of the chloroplast. So light-independent reaction. The principal role of a light-independent reaction is to reduce carbon dioxide to form carbohydrates. So this is to basically make that sugar. So if we think back to that really basic, simple overview of what photosynthesis is, it's to make that sugar. So step 1. Again, we're going to talk about it in one way and then show it in another way. So we have an enzyme. It's got a really complicated name, ribulose bisphosphate carboxylase. You'll see it written, I can guarantee, in pretty much every Calvin cycle diagram as Rubisco. Okay, so if you see Rubisco anywhere, it's ribulose bisphosphate carboxylase. Okay, and Rubisco triggers the carboxylation, so combining with CO2, so these might be words that you're sort of remember from chemistry, of ribulose bisphosphate, RUBP. and it's a 5-carbon compound. So if we look at this diagram, it will make more sense. So what happens is we have got RUBP, which is ribulose biphosphate, and that is a 5-carbon compound. Now you'll start to see that we start saying how many carbons are in each of these compounds, and we actually count the carbons of the cycle. And it combines RUBP, so rubisco is the enzyme, and it triggers that carboxylation of RUBP. So that is going to happen. Now, step 2. RUBP and CO2, they produce a really unstable 6-carbon molecule. And when literally it makes it and then it breaks apart straight away, and it breaks down immediately, and it forms 2 molecules. And it's 2 molecules of glycerate 3-phosphate. Again, on these Calvin cycle diagrams, you'll see glycerate 3-phosphate referred to as GP. We use a lot of shorthand for this part of the Calvin cycle, so it's good to know the full names because you will just see lots of initials all over the place. So yeah, RUBP is the enzyme rubisco carboxylizes RUBP. It forms an unstable 6-carbon molecule, which breaks down immediately to form 2 molecules of glycerate 3-phosphate. Now this is a 3-carbon molecule. So RUBP was a 5-carbon molecule. We've added a carbon from the carbon dioxide. It made that 6-carbon molecule, which breaks down immediately, and we now have 2 3-carbon molecules being formed. So step 2, that glycerate 3-phosphate, GP, is reduced, and it's reduced by that ATP that we have back from the light-dependent reaction, and it's reduced by the reduced NADP. So this forms 2 molecules of triose phosphate. If I draw this on, so basically these 2 things are happening at once. So we've now got those 2 molecules of glycerate 3-phosphate. And it's reduced first by the ATP. When it's reduced by the ATP, each molecule of glycerate 3-phosphate gains a phosphate. Therefore, each molecule of ATP loses a phosphate. So the ATP is yes, it is hydrolyzed. It's hydrolyzed to form ADP. It is also reduced by the reduced NADP. It doesn't have saying reduced twice, so NADPH. And the NADPH then obviously becomes NADP. So that happens. And when it's reduced by ATP and reduced NADP, oh, don't know, it forms 2 molecules of triose phosphate. And this is a 3-carbon molecule. So our numbers of carbons stay the same. So at this point, we're at 5, then we're at 6, we're at 6. Then our triose phosphate is a carbohydrate. So we can use, oh, I hate it moves so quick. And we can use a small amount, and a small amount of it is used to build other carbohydrates. So some of it actually, we lose some of it at this point of the Calvin cycle. So we can use it to build other carbohydrates such as glucose and starch. And this can also lead to the formation of other complex molecules further down the line, such as amino acids and lipids. So we're actually going to lose some of that triose phosphate at this point. But most of the triose phosphate is actually recycled, and we use it to make more RUBP. So now you can see how it's this cyclical process. So we use it to make more RUBP. To make more RUBP, ATP is converted to ADP and phosphate in this process. So we're losing that phosphate again. And so for every molecule of glucose made, 5 molecules of RUBP are produced. And that's a really, really important thing to remember. fact to remember. So for every single 1 molecule of glucose we make, 5 molecules of RUBP are produced. And basically this is a cycle that just keeps going on and on and on. So if the number of carbon atoms present in each compound is added to the diagram, we can see overall, like I was saying, that it balances. So we start off with 5 carbons, we add 1 carbon in, we have 6 carbons, we lose a carbon here to make those complex molecules, and then we go back to 5. So this is that process of, for every 1 glucose, we make 5 RUBP molecules. So products of the light independent reaction that pass back into the light dependent reaction, as we said earlier, are that reduced NAD. Products of the light independent reaction, so the one the Kelvin cycle just said, that actually go all the way back to the light dependent reaction are NADP, ADP, and those inorganic phosphates. So those NADPs, ADPs, and inorganic phosphates that we just said, oh yeah, they're there. And when they join, they make ATP or they make reduced NADP. Where do they come from? Well, they've come from the light independent reaction. And then products of the light independent reaction that are then used in other processes are that triose phosphate. And like we said, that's used to build complex carbohydrates, amino acids, and lipids, so things like glucose to start off with. So that triose phosphate, it's a 3-carbon sugar, and it can be used to make a huge amount of different complex molecules. So pairs of these TP molecules can combine, form hexose sugars such as glucose, and they could isomerize and become fructose. So you don't need to know this in super detail, but you need to know that they can become different complex biological molecules. So these monosaccharides can combine to form disaccharides, such as sucrose, and polysaccharides, such as cellulose and starch. And triose phosphate can be converted to glycerol, and this may be combined with fatty acids to make lipids. So the sequence of events, now let me just check you've got the right place of it starting. So ribisco, which is that enzyme, catalyzes the carboxylation of ribulose bisphosphate. Then what happens is an unstable 6-carbon compound is temporarily formed. That breaks down straight away. And when it breaks down straight away, it forms 2 molecules of glycerate 3-phosphate, and they are made there. Glycerate 3-phosphate is then reduced by ATP and NADPH. And when that happens, that forms 2 molecules of triose phosphate. Carbohydrates and other complex molecules are made, and triose phosphate is then used to form more ribulose bisphosphate. So, summary of photosynthesis as a whole. We have our light-dependent reactions happening in the thylakoid, and the light-independent reactions happening in the stroma. Light-independent reaction. Light-dependent reaction. Light has to hit the photosystems. Water is also present. We also have photolysis occurring. So light hits the photosystems. Water is split via photolysis. So we have oxygen being formed. Oxygen's a waste, and that goes out of the plant. Light-dependent reaction produces ATP, and that obviously makes ATP by that photophosphorylation of ADP and an organic phosphate. That ATP is passed on to the light-independent reaction. Light-dependent also forms NADPH, and that is passed on to the light-independent reaction as well. In the light-independent reaction, carbon dioxide is used to carboxylize RUBP. And the products of this, well, we get NADP, which passes back to the light-dependent reaction, and then ADP and an inorganic phosphate. And then another product, which is the main point of photosynthesis, are those carbohydrates and those other complex molecules. So, statements about photosynthesis, true or false. So during cyclic photophosphorylation, both photosystems 1 and 2 are involved. That's false. Only photosystem 1 can do cyclic photophosphorylation. ADP is formed as electrons are passed down chains of electron carriers in photophosphorylation. That's false. ADP and an inorganic phosphate bond during that to form ATP. The light-independent reactions take place in the stroma within chloroplasts, and that is true. So it's light-independent, no light's needed, happens in the stroma, which is great because that's full of all those enzymes, such as Rubisco, which we need. NADP is made in the light-dependent reaction and used in the light-independent reaction. That's false. It's the other way around. So remember, reduced NADP is made in the light-dependent reaction and is used in the light-independent reaction. And then in the light-independent reaction, NADP is made and then passed back to the light-dependent. ATP is made in the light-dependent reaction and used in the light-independent. That is true. So, just going to get Calvin cycle up one more time. I'm going to look at this one. If it's, yeah, I don't like that one, so I'm just going to draw one up really quickly and then we will finish. So, share whiteboard. So I just want to go through that light dependent, independent stage again, the Calvin cycle. So the products that we have coming into this are ATP and reduced NADP. So we're in the stroma, we have this enzyme called Rubisco. I'm going to put this in a box, a circle. And Rubisco catalyzes basically the carboxylation of RUBP, that's capital P, which is ribulose bisphosphate. So I'll pop that here. Ribulose bisphosphate. And it catalyzes the carboxylation of that. And obviously remember, carboxylation is the addition of CO2. So we've got our CO2 coming in here. So RUBP is a 5-carbon compound. CO2 is a 1-carbon compound. When they bond, they make a 6-carbon compound, which is carbon compound, which is really unstable, which breaks down immediately and forms 2 glycerate triphosphate molecules, which is 2GP. I'll put that here, glycerate 3-phosphate. Makes that there. Each one of these molecules is a 3-carbon molecule. Then these glycerate 3-phosphates are reduced. They're reduced by our ATP, which has come from our light dependent stage. It's 2 lots of ATP. And obviously that ATP is getting reduced by that, is going to become ADP, inorganic phosphate. And it's also reduced by our reduced NADP. Which will then just become NADP. When that happens, we then get 2 triose phosphate molecules, otherwise known as TP. These are 3-carbon compounds. And at this point, some of these go off, or one of them. So I'm going to put one here, 1C. One part of this triphosphate goes off to make useful organic substances, such as glucose. So then anything that's left goes back to form more RUBP. And to do that, it's reduced again by our ATP, which then is going to obviously produce ADP plus PI. And we go back to our 5-carbon compound. Now, this cycle happens 6 times. So I'm going to do this in a different colour, green, because I haven't used it. So this cycle happens 6 times. So in total, 6 carbon dioxides are used. So we have 6 carbons there in total. 12 of the GP compounds are going to be formed. So we'll have 36 carbons being present at this point. Down here, again, we'll have 12 of these, so it'll be 36 carbons. Here, 1 carbon goes off. So 6 carbons go off to make glucose. And then here, obviously, we'll have 6 lots of that, times 5 would equal 30. So again, it balances out the whole way round. And that is the Calvin cycle, which is our light independent stage of photosynthesis. So let me just stop recording.